8.2 Outside the mitochondria, glycolysis. Glycolysis, which takes place within the cytoplasm outside of the mitochondria, is the breakdown of glucose to 2-pyruvate molecules. Since glycolysis occurs universally in organisms, it is most likely evolved before the citric acid cycle and the electron transport chain. This may be why glycolysis occurs in the cytoplasm and does not require oxygen. There was no free oxygen in the early atmosphere of the Earth. Glycolysis is a long series of reactions, and just as you would expect from a metabolic pathway, each step has its own enzyme. The pathway can be conveniently divided into the energy investment step and the energy harvesting steps. During the energy investment step, ATP is used to jumpstart glycolysis. During the energy harvesting steps, more ATP is used than was to get started.
As Oxidation of G3P now occurs by the removal of electrons accompanied by hydrogen ions. In duplicate reactions, electrons are picked up by coenzyme NAD+, which becomes NADH.
When O2 energy is available, each NADH molecule will carry 2 high-energy electrons to the electron transport chain and become NAD+ again. Only a small amount of NAD+ is needed to be present in a cell because, like other coenzymes, it is used over and over again. The addition of inorganic phosphate result in a high-energy phosphate group per C3 molecule. These phosphate groups are used to synthesize 2-ATP. This is called substrate-level ATP synthesis, sometimes called substrate-level phosphorylation, because as an enzyme passes a high-energy phosphate to ADP, an ATP results. Notice that this is an example of coupling. An energy-releasing reaction is driving forward an energy-requiring reaction on the surface of the enzyme. Oxidation occurs again, but by the removal of water. Substrate-level ATP synthesis occurs again per C3 and 2 molecules of pyruvate result. Subtracting the 2 ATP that were used to get started, there's a net gain of 2 ATP from glycolysis.
During glycolysis, BPG is a 3-carbon substrate that gives up a phosphate group to ADP. Each gray ball is a carbon atom. This reaction occurs twice per glucose molecule.
Inputs and outputs of glycolysis. Altogether, the inputs and outputs of glycolysis are as follows. The inputs are glucose, 2 NAD+, 2 ATP, 4 ADP, and 4 phosphate, and the outputs are 2 pyruvate, 2 NADH, 2 ADP, and a 4 ATP total for a 2 ATP net gain. Notice that so far we have accounted for only 2 out of a possible 36 or 38 ATP per glucose when completely broken down to CO2 and water. When O2 is available, the end product of glycolysis, pyruvate, enters a mitochondria where it is metabolized. If O2 is not available, fermentation, which is discussed next, will occur.
First, 2 ATP are added. Second, 2 NADH are produced. Next, 4 ATP are produced by substrate-level phosphorylation. Last, 2 pyruvate are formed.
Basically, all we need to know about glycolysis is that it's a 10-step process that breaks down one glucose molecule into 2 3-carbon pyruvates or pyruvic acids, and that also releases 4 ATP. And this process does not need oxygen, and each step catalyzes a different enzyme, which produces raw material for the Krebs cycle, which is needed for aerobic respiration. Also included in glycolysis is an enzyme called phosphofructinase, or PFK, which inhibits glycolysis when there is enough ATP.